Suppose you have some cells in lab that you want to know their numbers. One thing that you can do is you can use a hemocytometer. A hemocytometer is a microscopic slide like this one, and it has two parts where you can put your cells and check how many numbers you have got. Just like in a microscopic slide, you put your cells in this space over here, and then on top of that, you cover it with a cover glass, or you can call it also a cover slip. So it creates a defined compartment where cells can sit. After that, you take this microscopic slide under microscope and then focus on one of the two grids. Now, when you look closely at the hemocytometer's grid, what you can see is 9 squares. Now, out of these 9 squares, if we went into more detail, what you are going to see is there are some corner squares over here, and there are 16 squares in the corners. Okay, so the corner squares have smaller 16 squares, whereas the middle one over here, which is mentioned as A, has 25 squares. Each of these squares hold 0.001 milliliter of the solution containing the cells, so that you know how much volume is being held by each square. Now, when you count your cells, let's say in this square over here, let's say you got 6. You know 0.001 milliliter, or also called zero point 0.1 microliter, has 6 cells. In this way, hemocytometer helps us to know how many cells are present in a defined volume of solution. Now, since we know how many cells are present in zero point 0.1 microliter, we can extrapolate it and know how many cells are present in 1 ml, and similarly, we can know how many cells are present in how mLs of solution we have. Now you can see over here the area of this square is also defined. Its area is 1 millimeter square over here, and let's move on and see how you count cells in a hemocytometer. In a hemocytometer, what you're going to do is when you're counting cells, if a cell touches these boundaries over here, you usually count the cells that touch two boundaries only, which is the top one and the right one. And you only take the cells that touch these boundaries into considerations, or the cells that are present in this area defined by the box, whereas the cells that lie on the left as well as the bottom boundary are not counted. You will know why it is done because it's done to avoid repetition, and I will show you this with an example. You can see over here I am starting to count my cells in one of the corner squares of the hemocytometer. As I move along, I'm only going to count cells that touch the top as well as the route boundary. Okay, so when I come into this box, I see one cell sitting over here, so I will write I have one cell in this small box. These are 16 total. As I move on to the next box, I see there are no cells in the boundary, whereas here at the bottom there is a cell sitting, but I will not count this because I'm only going to count the cells that touch this boundary or this boundary, which is the top and the right one for each square. I move on to the next square, there is no cell. Similarly, when I come over here to this cell, I see now I'm going to count the cell that touches the top as well as the right boundary. I see there is one cell that touches the top boundary, whereas one cell is touching the bottom boundary. For this cell again, I'm not going to count this because it is touching the bottom and or the left boundary. So I'll write one over here. I come over to this cell over here. This again is touching the left boundary. I'm not going to take it into consideration. Then we come over here. It is touching the right boundaries. So we will write one over here. And then we come back over here. There is one cell, two cell. I will write two over here. I'm not going to take it into consideration because it's touching the left and the bottom boundary. Then I move off to the next square over here. Now I'm going to take this into consideration. I have one cell in this box. Moving on to the next box, again I have one cell. So you can see if I had counted this cell over here into this box, it would have given me number 3. Whereas when I had moved on to the next one, I would have gotten confused if I had taken this cell into consideration when I were counting in this box, or should I write it over here. So this is why when you're counting in a hemocytometer, you only take 2 of the boundaries into consideration, the cells touching them, whereas the bottom and the left one belong to the next cell. Okay, so in this way I have counted all the cells that are present in one of the corner boxes, and similarly I will count in the rest of the corner squares of the hemocytometer. Now I have a equation, or you can say a question for you to solve. I will help you solve them. So it is, we have diluted a cell suspension up to with 1 part of our solution, and we added, let's say, 10 parts of water. Okay, so this right over here is how much dilution was done before you took your cells and put them into the hemocytometer. Okay, now they loaded it into a hemocytometer. You count the number of cells in the 4 large corner squares. Okay, so how many squares you counted? 4 over here. And what's the count you got? In one box you got 45, 50, in one you got 48 and 47. If we know how many total cells we got in the 4 large squares, it's going to be close to 190 over here. And then they ask us what is the cell concentration, and we have to tell the concentration in cells per ml in the original sample. Now in the original sample, which is going to be, we have to find the concentration, how many cells per ml they are, and also we have to take into consideration the dilution we had done before we took the sample, like we before we put it in the hemocytometer. So this right over here is our equation that we can use. So for cells per ml, it's going to be the total cells we counted, which is 190 over here, and the dilution factor, which is going to be 10 over here. So our dilution factor is going to be 10, and we will multiply it with the volume conversion factor. Now we know if we counted our cells in 1 square, it was going to be the number of cells present in 0.1 microliter or 0.001 ml of solution. When we multiply it with 10 raised to power 4, we can also write it like this, 10 raised to power 4. It will convert that amount into 1 ml, which is cells per ml. Okay, so below over here we will write the number of squares. So this thing over here is going to become the average cell count per square multiplied by the dilution factor and multiplied by the conversion factor, and then we'll see what we get. We put all the designated numbers, 190 for the total cell count. When we add all of these, we take 10, which was our dilution factor over here, how many times we had diluted our cell suspension before we introduced into the hemocytometer, and the volume conversion factor, which is 10 raised to power 4, and the number of cells we had counted in the in the number of squares we had counted the cells, which was the 4 corner squares. Okay, so we are going to get this number over here, this big huge number. So these are the number of cells per ml that are present in this question over here. Whatever kind of question you get, you can use this equation. You can find different number of things with this, the total cell count, number of squares, the dilution factor, cells per ml, which is going to be the cell concentration. So this was hemocytometer. I hope you liked it. This is Biopathway. Please subscribe to us, and if you like our channel, leave a comment. Thank you.